Good afternoon, students. Department of Histology and Embryology represents for you the lecture Nervous Tissue. Nervous tissue is one of the special tissue in human bodies which to provide the extra special functions like different with functions of tissue, of common tissues. Especially the structural base of this tissue is neuron, nerve cells. So we have to talk about the definition, nervous tissue. The nervous tissue is the system of specialized cells which can perceive irritation from various physical and chemical agents from external or internal environment. And transmit the resulting excitement to functional organs. The nervous tissue realizes the interconnection and regulation activity of tissues, organs, and their connection with the external environment. That is why the nervous tissue constitutes the structure organs of nervous system and play integrated role in all types of functional system in human body. The basic property of nervous tissue and excitability and conduction. Through the sensitive nerve ending, the organism obtains the information about an external world. Nerve tissue consists of neurons that transmitted electrochemical impulses and the supporting cells that surround them. It contains little extracellular material. The nervous tissue compound a basis of nervous system. Look at this schematic diagram. We have seen here the nervous tissue which divided on nerve cells, neurons, and neuroglia, neuroglial cell. By the way, as you have seen here, not present extracellular matrix. Why? Because that nervous tissue is the result of evolution of divergence, evolution of tissue. During this evolutionary transformation, the extracellular matrix disappeared and its replacement with specialized cells, neuroglial cells. A lot of types we will study today in this lecture. But the main structure of nervous tissue is nerve cells or neurons. So, thus we understood that a simple tissue in human body without extracellular matrix, this is nervous tissue. Neurons are specialized to receive, integrate, and transmit electrochemical messengers, which is also named neurotransmitters. And via this mechanism, it's to regulate the impulse activities in all body. Neuroglia or supporting cells give mechanical, nutrition, support to neurons. Neuroglial cells have secretory functions and isolate neurons and their processes. What about development? Overlying ectoderm forms neuroepithelium, which thickens and form the neural plate. As the marginal disc plate continued to thicken, the plate bulkmass forming the neural groove, with edges continue to grow towards each other until they come together, forming the neural tube. So we just have a little step. Next, look, we have seen here embryonic disc. In embryonic disc, we very good see cut edge of a neon, red color, neural folds, neural groove, and this structure is totally named neural plate. Look, at the lateral surface is very good visible somite. That's the first three portion of somites, three pair of somites, which is started the process of closing of neural plates into the neural tube. Somites, that's very important, the dorsal, paraxial, segmented mesoderm, which to provide the somatic periods of development. And the first step of this period, this is neurulation. Previous, we have seen primitive knot and primitive straight like components of last step of ingration or second stage of gastrulation. The main question is activities, interactions between neural ectoderm and mesoderm. Look at this one, we have to see that the neurulation is definition, process of formation of neural tube. The embryonal induction can be illustrated by the active production of signaling molecules from notochord into neural plate, which to transforming into the neural folds and neural groove. As a result of this is infoldings, invagination inside, then neural groove to continue deeper and deeper decrease, and one moment to form the neural crest cells like a tip of the neural folds, and this is neural crest in future will give rise formation of ganglionic plates or system of neural crest cells which migrated for the formation of peripheral nervous system, nerves, and we have discussed this question on the next slide. Look at this slide again, and to remember, first of all, that's neurulation, process of formation of neural tube, which is stimulated from the signaling molecules from notochord, and without this is activation, continued the process of formation of neural tube. Next moment, this is derivatives of neuroectoderm. We have seen here the picture of embryo, fetus. Sorry, this is the fetus, and we have the time of histogenesis. We have seen here the formation of pharynx, pharyngeal pouches, pharyngeal clefts, head. We have seen the limb and tail, and also we have the process of segmentation of mesoderm and formation of body cavities, and the main idea that's ectoderm to give rise to formation of future nerve system and sensory organs. This is the first component, neural tube to form central nervous system. Neural crest, peripheral nervous system, and placodes, this is a thickening of surface ectoderm to take an active part in the formation of sensory organs with a combination with neural tube and neural crest. So neurulation started at 18th day of prenatal development, and we have seen the neural plate coloring blue color. Future neural crest cells is visible here like purple color. Also we have seen prechordal plate. This is a chorda mesoderm, not a chord mesoderm rudiments, which to provide the induction for the formation of neural tube. In the next slide we have seen very good visible neural groove, neural fold, and endoderm, mesoderm with somites, and splanchnopleurotic mesoderm and coelomic area coelom between the parietal and visceral layer of mesoderm. 22nd day of development, the somites start to press the neural tube, so neural plate from the lateral surfaces in the region of neck, and we have seen the wave of neurulation. We can be named that's cranial neurulation or cephalic neurulation wave and caudal neurulation wave. During these waves, that's somatic mesoderm starts to develop and step by step close the neural tube. Effect of it, we have the only two spaces which is opening the internal surface of neural tube. This is cranial neuropore and caudal neuropore. And neural tube in this stage ordinary full developed. One moment in 22nd day, as you remember our previous lecture in biology, start the heart beating. So this is very important critical period of development. Next, structure of developing neural tube. That the neural tube is formed, it can be classified on a three layer. Ependymal layer in simply with a mitotic division of cells turn to the cells of the cylindrical form and cover the central canal of spinal cord. We have very good thing here. Internal concentric area or ventricular area is also named ventricular with dividing cells. Back. Mentile layer consists of mitotic dividing neuroblast and spongioblasts, gradually turning into the mature neurons and neuroglial cells. So again, what is it? This one is a mentile area, whereas we have seen that migration of the cells from the germinal layer or ventricular layer and start to differentiate it from stages of formation. Bipolar neurons, pseudo-unipolar neurons, multipolar neurons, as this is very good visible here. So next is marginal layer. Is formed by processes of cells of ependymal and mentile layers. Later is give rise the formation of white matter of central nervous system. Example, we have very good thing in the peripheral area. So again, ventricular layer with cylindrical shaped cells, mentile layer with the cells of differentiated, and the marginal layer with the processes of these cells. Spongioblasts give rise to formation of astrocytes and oligodendrocytes. In the light microscope, the first identification of differentiation of neurons, nervous cells, is appearance of thin fibrils in their cytoplasm. The body of the cell gains the piriform shape, and form its point, and the process an exome induces. Such cells are termed neuroblasts. They further turn to the neurons. Division of neuroblasts descended by mitosis. Look at this one. We have the derivatives of neural tube. Again, neural tube. Again, cylindrical shaped cells, and the shape will change the position of the structure of the body, cell body. And we have seen the neuroepithelium. How does it transform? At the periphery of this is neuroepithelium is very good visible mesenchyme. From this is mesenchyme. Ordinary mesenchyme is neurocrest origin, can be differentiated microglial cells, that's macrophages of nerve tissue. Nerve tissue macrophages is named microglial cells. In future, it's derived from origin from the premonocytes of blood in red bone marrow. Next, the classical cells of neural tube. This is neuroblastic lineage and glioblastic lineage. Neuroblastic lineage starts from the apolar neuroblasts, the cells round-shaped without processes. Second, bipolar neuroblast, only one processes, and one processes is more longer, another is more shorter. Next is unipolar neuroblast, when another second processes is degenerates. And after unipolar neuroblast, we have seen multipolar neuroblast. That's classical neurons in future. Parallel with the formation of lineage of neuroblast neurocyte, we have the glioblast or spongioblast lineage. In this case, spongioblast can be firstly disseparated from the neural epithelium, and the first is disseparated ependymal cells. Ependymal cells arise straightway from the ventricular layer or inner layer in synthesis, and to give rise to formation of cells which synthesize liquor. This is a liquor of central canal and ventricles, which to support, which is an internal environment of a central nervous system, and especially ventricles and central canal. The structure of ependymal cells we will discuss a little later, but look at this one. We have seen how the epithelium of choroid plexus also arises from this source. From another side, we very good seen how the glioblast, spongioblast dividing into the two lineages. The first lineage is named astroblast, which to give rise to formation of protoplasmic astrocytes, and the second lineage is fibrous astrocytes. That's a specialized glial cells for supporting the blood vessels in central and peripheral nervous system. One moment, oligodendroblast will give us formation of oligodendrocytes. Very interesting cells because they have been present for central and peripheral system, but in the central nervous system it's arise from neural tube, neuroepithelium, but in peripheral system is arise from mesenchyme of neural crest. Neural crest to give us information of this is Schwann cells especially. And form of development. That's when the nerve cells start to grow. It's first of all create axon buds, and this formation of a synaptic generation started at the terminal of these processes. Plasticity, that's a higher characteristic of the early neurogenesis. Plasticity is ability to form a new uh, terminals and to form a new synaptic junctions between the nerve cells and muscle, nerve cells and another nerve cells. And this is plasticity. It's highest in the first year of life. After that, we have seen that 75% of the neuroblast can die because is not be joined with target organ. Like example, target organ, this is a muscle, skeletal, striated skeletal muscle, fiber, for example. And also we have talking about cardiac muscle cells. We are talking about glands, maybe endocrine glands, which is also controlling by innervation. It's a formation of nerve ending. That's all. We have to know the law. If neuroblast is not be transformed to neurocytes with junctions with the target organ, like example, muscle cell, or maybe muscle fiber, or maybe epithelial cells, these neurocyte and neuroblast dying via apoptosis. Specialized radial glial cells to present in the wall of a neural tube for the regulation of migration of neuroblasts. Neuroblasts appear, as we're talking about, in the ventricular layer and start step by step upstairs in the upper layers. Sometimes can be migrated on the special processes of radial glial cells. So this is radial glial cells in future disappear. But another structure in 19 days of formation, very good visible under the electron scanning micrograph, we have seen the formation of neural crest. When the neural tube is closed, we have seen the cells extra portion of the neural tube start to migrate. That's the shape of the cells resemble on the stellate cells, or maybe like mesenchyme-like cells. And this is cells is named, population cells is named neural crest. Neural crest, or ganglionic plate, another name, it can be migrates into different areas. First of all, it's create dorsal root ganglia from the left and right side of the spinal cord. From another side, it's to form the synaptic trunk, sympathetic trunk, that's ganglions of celiac ganglion, renal ganglion, so extramural and intramural ganglion. Like example, intestinal tract, that's ganglions occupies the wall of an organ. In this case, is named intramural ganglions. One moment, that's neural crest population cells can be to give rise to formation of spinal nerve and skull nerve also. And we have to see that specialized neurons, like two types of neurons. This is pseudo-unipolar neurons of dorsal root ganglia, with satellite cells which also arise from neural crest. Schwann cells also provide the formation of myelinated nerve fibers, and of course multipolar neurons of sympathetic ganglia. So look, it's a very interesting example of histological divergence of cells and tissues type. As example, we have seen that neural tube consists from three layers: ventricular or incentric, mantile, and marginal. But these three layers can give rise to formation of different types of cells in central nervous system, especially spinal cord and brain. But from another side, that neural crest cells also divide on different population cells. One of them to transform into the neurons, pseudo-unipolar, another multipolar, one in the dorsal root ganglion, another in the sympathetic ganglion, and some cells will give us information of Schwann cells, and specialized type of cells to create the chromaffin cells in suprarenal gland medulla. The last moment, that's melanocytes. Melanocytes are specialized cells, pigment cells, which also developed from the neural crest. In this next slide, very good visible the position, localization of the neural crest, and one moment we have seen the migration of neural crest and how does it create some ganglia, enteric ganglia, preaortic ganglia. In one moment, in upper portion, we very good seen the formation of head and pharynx of the embryo. And this is very important moment that the skeleton of head, skull, to arise from ectomesenchyme. This is ectomesenchyme also developed from neural crest. Thank you for this part, and just we talking about neurons. Neurons are specialized to receive, integrate, and transmit electrochemical messages to another neurons, muscles, and secretory cells. Each neuron has a cell body, also called the soma, or another name pericardium, around the nucleus, comprising the nucleus and the surrounding cytoplasm and plasma membrane. Each neuron has a variable number of dendrites, cytoplasmic processes that collect incoming messengers and carry them toward the soma. And single exon, cytoplasmic processes that transmits messages to the target cell or target organ. The cell body, soma, pericardium, is the neuron synthetic and trophic center. The nucleus typically is a large, central, and euchromatic. It has prominent nucleolus and heterochromatin around the nuclear envelope's inner surface. The cytoplasm of the soma contains many organelles, including mitochondria, lysosome, centrioles, and of course rough endoplasmic reticulum. In the classical picture, in all anatomical and histological book is very good seen. That's a pericardium or soma contain nucleus and a lot of processes surround. Let's say dendrites. Dendrites is a short processes, but it's collected the nerve impulses into the soma. But that's only special process, which is named exon or neurites, has a special exon hillock. That's the area where the impulse was generated, chemically generated. And we have to see that the exon characterize the myelinated shift. And this is area which is covering by myelin sheath disappear by the special knot of Ranvier. We will discuss what is the knot of Ranvier, but that's a special adapted area for transmission of impulses. That the impulses is jumping from one knot of Ranvier to another, because in the area of myelin sheath it is impossible. And the terminal of axon to contain a lot of synapses, specialized nerve junction. The abundant free and rough endoplasmic reticulum associated polyribosomes appear as clumps of basophilic material collectively called Nissl bodies. The Golgi complex, which is well developed, packages the neurotransmitters in the neurosecretory or synaptic vesicles. Once packages, the vesicles are transported by the molecular motor proteins down the axon to the terminal bottom. That's we very have good seen in this picture. The first slide was stained by toluidine blue by Nissl, and we have seen Nissl bodies inside neurons. Compare that in neuroglial cells is not be visible any such a structure, is not be formed like specialized organelles. So this is Nissl body. It's a rough endoplasmic reticulum that was been specialized for the synthesis of neurotransmitters. Of course, in the neuroglial cells, that function is absent. They can't be synthesized any neurotransmitters or proteins for transmit the information or impulses. Like in other, we have very good seen the position of nucleus, position of Nissl body, localization inside the pericardium. Compare that in the stain silver impregnation with a combination of contrast stain hematoxylin, also very good visible glial cells, purple color, and neurites and dendrites of the neurons is also good stain with a special methodics. So this is some special methods can be to identified the organelles of neurons. One of the main organelles of neurons, this is neurotubules and neurofilaments. Both together is named neurofibrils. Also, we have seen that each neuron contains big, pale, round-shaped nuclei with a permanent nucleolus. Also, we have seen the numerous rough endoplasmic reticulum, especially this structure can be named the Nissl bodies, the numerous mitochondria. So neurons is very energetic cells because it need a lot of energy for synthesis of neurotransmitters, biosynthetical activities, protein synthesis, and Golgi apparatus, dictyosome. And also we have seen a lot of ribosomes also and some transporting vesicles. A lot of these vesicles accumulated in axon hillock. So dendrites also has vesicles, but without neurotubules and neurofilaments like in axon, in axonal hillock. Because that we also have to know that during this is metabolic exchange in the cell body of neurons can be identified lipofuscin. Lipofuscin is a yellowish-brown pigment granule is thought to be the remnant of lysosomal enzymatic activity. Lipofuscin granules increases with advancing age and may even crown the organelles and nucleus to one side in the cell, possibility affecting cellular function. Like example, we have seen it here. Look, this is a nucleus, nucleolus, nucleus, nucleolus. At the periphery in the cytoplasm of the perikaryon is very good visible yellow-brownish pigment lipofuscin. That pigment is ordinary primary appear in the dorsal root ganglia, in sympathetic ganglions, around 22-25 years old, but is not visible in that time in the brain, but with the time it can be appear in the brain also. So, also we have seen the characteristic of lipofuscin. That's the lipofuscin is result of transformation of lysosome and mitochondria during the metabolic exchange with the age. And another very important structure is neurotubules and neurofilaments. Okay, I'm back, my dear students, and we continue our lecture. So now, please be attentive, and we have to see a very important moment of structure of our nerve cells. We have to continue for one moment again. Axon is one of each neuron, and this is processes of neuron which to carry the information from the cell body to periphery. An axon is divisible into the several regions: axonal hillock, initial segment, axonal proper, terminal arborization, and terminal end bulb, terminal bottom. Some axons have branches, termed collaterals, is named collaterals. But a little back for our talking of identification of neurons under the microscopic technique. I am repeat the information. That's Ramon Santiago and Cajal, famous Spanish scientist, parallel with the famous Italian scientist Camilla Golgi, to provide the using for identification of nervous cells and neurons, especially silver impregnation technique. The silver impregnation technique is used the nitrite of silver, and nitrite of silver can be absorbed on the surface of the fibers. Especially these fibers is neurofilaments and neurotubules, and that is very famous, known like neurofibrils, which you have to drawing in practical classes. This is like a meshwork. This is meshwork of neurofilaments and neurotubules. It's not the original, it's like effect of staining, because the neurotubules and neurofilaments located in the different levels of the cells. One moment, as Camilla Golgi to identify the Golgi apparatus. This is the dictyosomes. Dictyosomes is very good visible with silver impregnation also in the dorsal root ganglion cells, pseudounipolar neurons. Santiago, Ramon, and Cajal can be identified neurofilaments and neurotubules, which is named neurofibrils. So back for our discussion, next method is a gold impregnation. With a gold impregnation, Santiago, Ramon, and Cajal to have been confirmed the existence of synaptic bulb. That's each neurons to communicate with another neurons with special axonal terminals and to form the special synaptic junctions, synapses. And we have very good seen with this is gold impregnation. In this case, we use the chloride of aurum fluoride, gold chloride, and this is effect to very beautiful, is visible under the microscope. From another side, we have seen that ordinary hematoxylin and eosin stain is not be effective in this variant, not be visible neurofibrils, not be visible Golgi apparatus, and of course absolutely not visible some specialized synaptic junction. Well, so back for our modern techniques, this is identification of culture of neuroblast in vitro investigations. In vitro investigations of specialized cholinergic basal ganglions, we have to identify special proteins nestin. Nestin is a neurofilament. Neurofibrils contain nestin. It's an intermediate filament of nerve cells and to very good identified with immunofluorescent method, immunofluorescence. So next, talking about axonal transport. Another name, exoplasmic, correct, axonal transport. The movement of metabolic products through the exoplasm, which can be fast, 400 millimeter per day, or slow, 1 millimeter per day, involves neurotubules and neurofilaments. We have to identify it like anterograde transport or orthograde exoplasmic transport, moves newly synthesized products toward the axon's terminals, herborization, and can be fast or slow. So this is movement of neurotransmitters, specialized proteins, neuroproteins, which to migrated into the synapses from cell body into the terminal of axon. But retrograde exoplasmic transport, they return to warm materials to the pericardium for degradation or reutilization is usually fast. So remember, exoplasmic transport, axonal transport can be anterograde and retrograde. Anterograde is a fast variant and slow variant, but retrograde transport ordinarily fast. We have to see it in this unical diagram. We have seen the soma, pericardium, axon, axonal terminals, we have seen Golgi apparatus, rough endoplasmic reticulum, and the secretory vesicles. This is secretory vesicles, is vesicles which to move anterograde transport into the axonal, in the region of the axonal terminals, where is to form synaptic bulb. And we have how is these vessels, this is vesicles, is released from the presynaptic membrane into the synaptic clip. And opposite, we see how the this is vesicles pinocytotic vesicles is migrated from the presynaptic membrane into the axonal processes. So the form of developing was discussed, and now we have continued that philopodia formation in axons and actin network formation. So when terminals of axons start to develop, it's to forms the many processes and many sub cytoplasmic protrusions. This is protrusions is named philopodia, which is more longer, and the lamellapodia, which is more smaller. And we have seen a system of actin networks which to provide the very high level of plasticity modifications of the axonal terminals. And sometimes the people asking, what's the function of this is plasticity? Its ability for transformate the junctions between different types of neuronal cells. And we have two neuronal networks, and this is neuronal networks. It's not stability, it's a mobile system which is all the time adapted for the exchanges in the internal or external environment. And if we have talking about the main process which take place in between exoplasm and system of the nerve tissue, this is of course generation of neuronal impulses. We was discussed the question about axonal hillock, as you remember. In axonal hillock is also present the area where is to generates the impulses via the migration of sodium and potassium ions through the plasma membrane. As you have seen that intracellular fluid accumulate more sodium and release less potassium. So 3 sodium and 2 potassium. This is a balance. This was being controlled by sodium potassium ATP ACE pump. Effect of it, we have the generation of impulse activities, which is regulating via the migration of sodium and potassium ions, as we have seen here. More effectively you will study in physiology, but today we need to remember that's the potential of action. This is the area when the membrane will be change the electrical voltage. And example, we have the 3 conditions. That's resting potential, which is not active, where is the area of minus, and depolarization, when that's the sodium ions is more active, and repolarization, when we'll have to change again. So from resting potential to depolarization and to repolarization. In this case, we have how the impulse migrates in the area of node of Ranvier. This is unmyelinated area is named node of Ranvier. And saltatorically, that like is jumping because a salto is a Latin word, like jumping from one node of Ranvier to another node of Ranvier, and it's very fast to migrate from side by side, especially in myelinated nerve fibers. Morphological classification of neurons. We have sometimes 1, 2, 3, 4 types of neurons. Unipolar neurons possesses only one process. Bipolar neuron possesses two processes. Pseudo-unipolar neurons develop from the embryonic bipolar neurons. Those processes migrate around the cell's body during development and eventually fuse into the single process. Multipolar neurons, the most common type, possess various of arrangement of multiple dendrites emerging from the soma. And we have to see the examples. That's apolar neurons is really not present in humans, is maybe only in embryo. Unipolar neurons present only in development but not present in nature in adult organisms. But bipolar neurons, this one is bipolar neurons, is the classical neurons, which is present in any organs, sensory organs. And we have the pseudo-unipolar neurons, which is ordinarily present in dorsal root ganglion. It's one of the sensory neurons in dorsal root ganglion. And multipolar neurons, the main type of neurons. Again, watching that in bipolar neurons we have two processes, dendrite and axon. Pseudo-unipolar neurons, T-shaped, dividing one process to give rise to formation of dendritic branches and axonal. And we have also the classical neurons, which we studied a minute ago, with dendrites surround the cell body and one long axon. So examples of these neurons very good studied by the Russian histologist and one of the famous histologists in Russia and also for investigations of the brain, Vladimir Bets. He was being the professor of Kiev's, the histology department in Kiev in the University of St. Vladimir. That was happened at the end of 19th, beginning of 20th century. So we have the unique structure of these cells. One of the famous of it is named Betz cells. This is pyramidal neurons of a cerebral cortex. That's neurons to operate the movement. This is motor neurons, and a lot of them too can be identified like a system of processes around the cell body, but the axons is started from the center. This is trigonal pyramidal shaped neurons. And this is the largest cell in a human body. Diameter of the cell 150 microns, and the length of this axonal process is sometimes more than 1.5 meter. From another side, we have seen pseudo-unipolar neurons of receptorical arc. This is neurons which is located in the dorsal root ganglion. And also we have seen another multipolar neurons and bipolar neurons of olfactory zone. This is olfactory neuronal cells. Okay, next functional classification. According to the functions, it's a classification basics on the reflex arch. We have talking about sensory neurons, afferent neurons, receive sensory input at their dendritic terminals and conduct the impulses to the central nervous system for processing. Motor neurons or afferent neurons originate in the central nervous system and conduct their impulses to muscle, gland, and other neurons. Interneurons or associated neurons, located completely in the central nervous system, functions an interconnector of integrator that establish the networks of neuronal circuits between sensory and motor neurons and other interneurons. What's the main idea? This is, of course, the structure of reflex arch. This reflex arch underlie the activity of the nervous system, That is a pathway where the nerve impulse migrated for the providing the motor functions and sensory functions and secretory functions. And the different type of reflex we know is like one of them is basically orientated for the activities of the human bodies. They represent a chain of neurons which provide responses of organs and targets. It's rapidly to receptors stimulation. In reflex arch neurons, the bound of other synapses form three links. First, this is a central portion of reflex arch. Second, this is receptors, efferent neurons, motor, efferent neurons, possessed between them associative interneuron, and the elementary variant of arc interneurons can be mixed. Example of reflex arch we just very fast to discuss. So first of all, this is a central portion of reflex arch. This is a spinal cord gray matter. In the gray matter we have seen dorsal root and ventral horn. Dorsal horn and ventral horn. We have seen that very interesting that except the central portion, we're talking about sensory ganglion, ordinary sensory ganglion located in the dorsal root ganglion. One moment we're talking about also receptors. Receptors in an effector. So we have seen the pain reflex is visible here from the left, and ordinary this is somatic reflex, and this is from left is a visceral reflex, somatic reflex and visceral reflex. In different cases, the impulse activities from the receptor migrated into the sensory nerve, into the ventral root. Ordinary, maybe we have talking about some dorsal root ganglion, and this is dorsal root ganglion to have the transmission of these impulses into the dorsal horn, in the interneurons around the area of lateral horn. Interneurons to provide association, the analyzing the information, and after that transmit into the motor neurons in ventral horn. Motor neurons to activate the spinal nerve, and effect of it is to activate the movement of the finger. One moment another, that's dorsal horn also communicates with the dorsal root ganglions in the case of visceral reflex. But in visceral reflex is very good visible that's in sympathetic chain ganglion, the motor neurons located outside the spinal cord. Again, receptor located inside the body, but to join with preventral ganglion. After that is migrated to the dorsal root ganglion. Dorsal root ganglion migrates the sensory neurons to migrate the transmit impulses into the interneurons in thoracic region of spinal cord, and from this is lateral horn, it migrates the transmitted impulses into the motor neurons in sympathetic ganglion. Thus, we understood that main differences between somatic reflex and visceral reflex, the localization of motor neurons. In somatic reflex, it's located in the ventral horn. In the visceral reflex, it's located outside in the sympathetic chain ganglion. Well, and the same we have seen in this schematic diagram, spinal cord. We have seen sensory neurons, this is spinal ganglion. Next is the ordinary somatic motor neurons. If we're talking about double neuron chain. And we have seen 1, 2, 3 chain, but in autonomic motor neurons in outside the spinal cord. This is a classical visceral reflex. During this is combination of the neuronal networks formation, we have seen efferent neurons, interneurons, efferent neurons, system of providing the localization, the cell body of interneurons in central nervous system forever. Motor neurons can be present or in central nervous system or in peripheral if we're talking about autonomic nervous system. Let's now discuss the ability of nerve cells to transmit impulses through synapses. So, synapses are specialized junctions by means of which stimuli are transmitted from neurons to its target cells. Synapses are named according to the structure they connect. Example, exodendritic synapses, with the axon joined with dendrites. Exosomatic, if the axon joined with soma, pericardium. Exoaxonic, if axon joined with axon. Dendritic, if a dendrite is joined with dendrite. Look at this one, types of synapses. First of all, look, we have seen the presynaptic area and postsynaptic area, and if this is axons, but we have seen here different combinations. Maybe this one is not hilo because this is maybe dendrite, so dendritic synapses. This one we have seen axon because the hillock is present, and we have seen the exosomatic synapses. Exosomatic synapses. And here we have seen maybe dendritic synapses and some variant of joint with these synapses. Well, three major structural components exist in each synapses. This is a presynaptic membrane. Look at this one. We have seen very good visible in synaptic bulb. Also, postsynaptic membrane with a special receptors, integral proteins. Also, we have seen the synaptic cleft, and presynaptic membrane is the part of the terminal bottom membrane closest to the target cells. In response of stimulation, neurosecretory vesicles in the bottom fuse with the presynaptic membrane and exocytosis of their neurotransmitters into synaptic cleft. Look at this very beautiful picture. We have seen how the neurotubules support the special secretory vesicles. Secretory vesicles migrate into the synaptic bulb. Here we have seen that's ions of sodium ions to stimulate the activation of calcium ions. In this case, that's a synaptic vesicles. This vesicles is named synaptic, that is named transporting neurotransmitters vesicles. This is synaptic vesicles. They are aggregates and start to activate with acetylcholine plus coenzyme A, and we have two aggregation of these vesicles with the calcium ions, and this is stimulate the exocytosis in presynaptic membrane, exocytosis. When this vesicle is exocytotical active, that's the neurotransmitters, acetylcholine, it's migrate into the synaptic cleft. Each molecule of acetylcholine occupies the receptors, in this case, to stimulate the migration of sodium ions. Sodium ions migrate through the center of this receptor if acetylcholine joins. So you understood that when the quantities of this acetylcholine are low, we started the activities of acetylcholine esterase, special enzyme, which is break down the acetylcholine. After that, the some portion of acetylcholine which is not used start the backside via the pinocytosis and provide the retrogradic transport, axonal transport. And this is very important moment because that's anesthesia using the special alkaloid, like example morphines, maybe like example some artificial blockage substances, which is ordinary blockage the acetylcholine receptors. In this case, when acetylcholine come into these spaces which is named synaptic cleft, it can be joined with receptor and affect the wave of sodium ions is not being transmitted. In this case, the organism will start like special hallucination, like a narcotic dream, and not be active this time, but is not be feel the pain and not be feel the some external environment, and can be live in inner condition of these cells. But of course, in this case, that's plant alkaloid like morphine, cannabine, and other can be destroyed with a special enzymes like acetylcholine esterase, because it's very dangerous for nerve cells, because they stop the blockage, the anterogradic transport and retrogradic transport. As a effect of these disorders take place the dimension that the nerve cells can die via apoptosis after time. Because it's very dangerous to use these drugs, anesthetic substances for blockage this transmission. There are perhaps 100 known neurotransmitters are neuromodulators is a known. Like this is a special name. Small molecules transmitters, acetylcholine, amino acid, glutamate, aspartate, glycine, GABA, biogenic amines. All these substances is ordinary neurotransmitters, like example catecholamines, dopamine, norepinephrine, noradrenaline, epinephrine, and some other which used for the types of neurons. Example, we have seen the substances is get different diameter of granules. Or another name is synaptic vesicles. Correct name, synaptic vesicles. So in the motor bulb, which is also named neuromotor junction, this is using this synapse, neuromuscular synapse, using the acetylcholine. But we just have seen it like this structure and under the microscope, and just we talking about supporting cells. The supporting cells is the central nervous system, other macroglia, astrocytes and oligodendrocytes and microglia, and ependymal cells. So first of all, we have seen all these cells, and this is diagram, microglia, neurons with oligodendrocytes, blue color, light blue color, green color, this is astrocytes, surround the capillary, ependymal cells. So let's now talk in ependymal cells. They derive from ciliated neuroepithelial cells lining the neural tube. Line the neural tube derivatives their veins, ventricles, and aqueducts, and the spinal central canal, and adult they retain their epithelial nature and some cilia. The lining resembles a simple columnar epithelium, but ependymal cells have basal cells processes, which is started from the center of the cells, and extending deep into the gray matter, like is very good visible here. So higher magnification shown for us, which is glial cells, very resemble of epithelial cells. Next moment, we have seen the schematic diagram, artistic work, and continue to astrocytes. That's astrocytes. We can classify it in protoplasmic astrocytes or mosid cells. Astroplasmic astrocytes, they have ample granular cytoplasm and short, thick, highly branching processes. Fibrous astrocytes are more common and white matter, silver stains reveal fibrous material and their cytoplasm. Their long, thin processes are less branches than those in protoplasmic astrocytes. From another side, we have compared fibrous astrocytes and protoplasmic astrocytes. As you have seen, fibrous astrocytes are not joined with peripheral myelinated nerve fibers. So we understood that the protoplasmic astrocytes is present in the central nervous system, but the fibrous astrocytes is dominated in peripheral nervous system. And we have to see it also here, very beautiful structure. How does it located these cells? Look at this oligodendrocytes, the same as like Schwann cells, very resembling. We have seen also that astrocytes to form the perivascular feed and to take an active part in the brain vessel barrier. And this is barrier we will study in the next lecture about the central nervous system. Processes surround the capillaries of the pia mater and the important component of the blood-brain barrier, astrocyte in the brain. And next we have seen this is meninges of the brain. We have seen this is a vessel and the position of this localization of astrocytes. Astrocyte is a green color, neurons in the purple color in here. Also, we have seen the component of this is barrier, but we will study more in practical classes. Look at this one. It's very important. The component is started from endothelial cells of blood capillary, next basal lamina, next this is tight junctions between the capillary and foot of processes of astrocytes. Next is this is cytoplasm of astrocytes, and after that we start the brain cells. Occur in both gray and white matter, like in Schwann cells of the peripheral nervous system, oligodendrocytes from the myelin, and occur in rows of myelinated entire axons. Unlike as the Schwann cells, each may provide myelin for segments of several axons. Microglia. Microglia, the smallest and rarest of the glia, occur in both gray and white matter. Microglial cells may derive from mesenchyme or are components of the mononuclear phagocyte system. You remember about the macrophages, and this is the microglial cells is macrophage of the brain, have phagocytotic capabilities. Look, this is very active, immunoreactive microglia, which is provide the phagocytosis, and it also can be divided on the processes and so on. Also, we have supporting the cells of peripheral nervous system. Schwann cells are the supporting cells of peripheral nervous fibers. The Schwann cells may envelop segmentation of several unmyelinated axons and provide the segments of single myelinated axon with its myelin sheath. That's satellite cells are specialized Schwann cells in craniospinal and autonomic ganglia, where they form the one cell thick covering over the cells bodies of the neurons, ganglion cells. Nerve fibers, unmyelinated nerve fibers and myelinated nerve fibers. Of course, myelinated nerve fibers is creation which developed like combination of axons ordinary and Schwann cells surrounding. We have seen the first of all, just talking about unmyelinated nerve fiber. The Schwann cells are elongated in parallel to the long axis of the axons, and the axon feed into the grooves in the surface of the cells. The lips of the groove may be open or closed, forming the mesaxon. Mesaxon, this is like invagination of Schwann cells, which took embedded inside this invagination the axonal processes. And large Schwann cells in the peripheral nervous system may have 20 or more grooves, each contain one or more axons. So this is grooves. Original is named mesaxon. Mesaxon. Look, this one is a mesaxon, like grooves, which surrounding is present the axons, and Schwann cells nuclei in the center. Like example, electronic microscopic microgram, we have to show also if we have Schwann cells nuclei and two axons, central cylinder, we have talking about unmyelinated nerve fiber. In myelinated nerve fiber, that present the peripheral and central nerve system, one Schwann cells provide a segment of a single myelinated axon with its myelin sheath. And external to the continuous with the myelin sheath, a thin layers of Schwann cells cytoplasm called the sheath of Schwann and neurolemma. This layer contains the nucleus and most of the organelles of the Schwann cells. As example, we have seen that's like layer by layer to surrounding the axon. The myelin sheath is segmented because it's formed by numerous Schwann cells arrayed sequentially along the axon. The junction wherever the adjacent Schwann cells meet in the void of the myelin. This site is called node of Ranvier. We have seen it here. And also we have seen that's where for the myelin between two sequential nodes of Ranvier is called internodal segment. Look, if we have seen one Schwann cells nuclei and one axon, this is myelinated nerve fiber, especially with more dark electron dense area. Myelination. The myelin sheath is composed of multiple layers of Schwann cells membrane. More effectively is visible like special animation which is present in this scheme. We have seen how the Schwann cells to spiralize during the axon, central cylinder, spiralized. As effect of it, we're talking about myelinated nerve fiber. That's in central nervous system, it's very good developed. Well, we just to continue our talking about this structure, and we completed our discussion. Nervous ending you study in the practical classes. Sorry, because the lecture is finished. Thank you for attention, and my best wishes to your study histology in this semester. So students, goodbye. Be healthy. Take care.